us. This is very special. Today we have a fantastic clinician scientist, Jim Handa. And I don't know Jim too well, but I spent once a little time with him in Kentucky. Jay and Buddy invited us. We went to this little hotel, and there is a picture of horses everywhere, on every corner. And that wasn't interesting for Jim. He wanted to go to bar. And then he was really, really surprised how many bourbons they have. And, after a while, he was like, oh my God, and this one, and this one, and this one. He went for all of them. And I would just maybe stop here. Or maybe I will tell you one more thing, what he did. After that, he was talking to the horse on the wall, and then police show up. But that's how it was.
Good job, Jim. We'll keep you out of Scotland, though, because that can be very dangerous for you.
All right, we have Jennings to tell the real story.
Yes, good morning, everybody. My name is Jennings Liu, and I'm an MD PhD candidate training in Dr. Palczewski's lab. Today, it's my distinct pleasure to introduce our keynote speaker, the Robert Bond Welch Professor of Ophthalmology and Chief of the Retina Division in the Wilmer Eye Institute at the Johns Hopkins University School of Medicine. He earned his undergraduate degree in biology from Brown University prior to obtaining his medical degree from the University of Pennsylvania School of Medicine. He then chose to stay in the city of brotherly love, go Eagles, where he completed his residency training in ophthalmology at the Wills Eye Hospital. Subsequently, he completed 2 fellowships, one in vitreoretinal diseases at the Duke University Eye Center and the other in intraocular oncology at the University of Southern California Children's Hospital of Los Angeles. As a clinician scientist, he specializes in medical and surgical management of complex vitreoretinal diseases such as age-related macular degeneration, diabetic retinopathy, retinal detachment, and retinopathy of prematurity. He also has expertise in managing patients with choroidal melanomas and metastatic tumors of the eye. Meanwhile, his research efforts have focused on the early causes of AMD, using molecular pathological approaches to understand how the eye transforms from normal aging to early disease. Over the course of his career, he has served on numerous scientific advisory and editorial boards, and he has authored over 200 peer-reviewed articles and is an inventor of over a dozen patents. Today, we have the privilege of hearing about his latest work on oxidative stress and the role of mitochondrial dysfunction in retinal degeneration. So without further ado, ladies and gentlemen, I present to you Dr. James Handa.
Jennings, thank you very much. I liked your introduction more than Chris's, but I must say that there is striking truth to what Chris said, and except for the past thing about me talking to the horse. So it was really a fun weekend that we had, or a fun couple of days that we had in Kentucky, and I think that's a time when I truly bonded and solidified my friendship with Chris. So it's true, Jenny, as Jenny mentioned, I'm going to talk about oxidative stress, induced mitochondrial dysfunction, and its role in RPE degeneration and macular degeneration. So just some housekeeping things. Here are my conflicts. Unlike most clinicians, I don't have a lot to declare. And then I really want to just sort of acknowledge some of the people, collaborators, and some of the people in my lab, particularly Marisol and Sayantan, who've done the lion's share of this work. And of course, the funding support to keep the lab alive, especially during the pandemic. So I really want to give a shout out with all seriousness to Chris and Grozny. I really appreciate giving the opportunity to talk at this session, and they really have made wonderful contributions. And the interesting thing is I was trying to dig up some dirt on Chris, so I went to Google, and the first thing that came up was his Wikipedia link. And everything was so complimentary, I really couldn't get through all of it. So anyhow, I thought I would acknowledge, I'm not sure how many people here have a Wikipedia page, but Chris does. So my particular interest has in the research line has been in early AMD, and we certainly know perfect vision with a normal macula. But one of the things that happens is oftentimes patients come in and they say, doctor, the prior doctor said my vision is perfect, but I feel like I'm blind. And when you measure their vision, it's under normal conditions, it's 20/20. But if you actually put them in low luminescence vision, darken the room, their vision drops to 20/80. So they clearly have some symptoms. And a lot of these early symptoms are related to decreased perifoveal rod dysfunction, such as reduced macular sensitivity, contrast sensitivity, delayed light exposure recovery, and delayed dark adaptation. And these are all, again, due to perifoveal rod dysfunction. And this correlates very nicely with Christine Curciel's work, really in the mid-1990s, where she showed that perifoveal rods were really the first cells to die. But along with that, there's work in this area, work in this region where the RPE has a higher probability of binucleate, 2 nuclei, and morphologic derangement. But also in the same time, some of the RPE cells are normal. So why does this happen? Why does the choriocapillaris capillaris die before the retinal pigment epithelium? It sort of points to the RPE. And so I've become interested in RPE heterogeneity, which has been well recognized, not necessarily described as heterogeneity, but you could certainly see the differences in the morphology. But there's not been a lot of interest, and I might add that's including a personal experience and study section. But if you look at cellular heterogeneity, ordered heterogeneity is protective, but degenerative heterogeneity due to aging tends to cause disease. Cancer, it's hotly studied, as well as in neurodegenerative diseases, and that's where a small number of cells can really drive disease. And I think the most vivid example is with metastasis. And so we've been asking the question, does RPE heterogeneity contribute to early macular degeneration? And certainly many people know that I've had a big interest in cigarette smoking's role in macular degeneration, because it is the strongest environmental risk factor for AMD. And if you look at smoke itself, there are nearly 5,000 toxins, and each puff of cigarette smoke has about 10 to the 17th free radicals. Obviously, not all of that gets to the eyeball, but it certainly elicits a pro-oxidative stress environment. And when we've exposed C57 black 6 mice to cigarette smoke and looked at the shape of the RPE cell, there's clear RPE heterogeneity and morphologic derangement that occurs in the animals that have been exposed to smoke. And we've previously shown, when we've looked at RPE cells exposed to cigarette smoke extract, that there's clear mitochondrial dysfunction, which is not, revolutionary. But when we compared it to other responses within the cell, like the unfolded protein response, it was really, the unfolded protein response was unaffected, which pointed us toward our interest in mitochondria. And certainly from trying to relate it to early AMD from our immunohistochemical studies, we've seen that TOM 20 immunolabeling is decreased over in areas where the RPE is dysmorphic relative to healthier RPE cells within the same eye. And then we can subtract out the melanin pigment to see the labeling pattern a little bit better. Well, certainly coming here, we know with Chris Kenny's work in the role of mitochondria in age-related macular degeneration, and this is one of her papers from 2010, but subsequently she's had 38 publications on mitochondrial abnormalities in the RPE as it relates to AMD. So I guess I'm preaching to the choir here in that sense. And certainly Deb Farrington's lab has spent a lot of time looking at the role of mitochondrial dysfunction, and she's identified mitochondrial damage in early AMD in the RPE, but not in the retina, which points again to a role for the RPE. And in terms of looking at oxidative stress and protection against smoking, we've been interested in NRF2. And for people who don't know, NRF2 is normally sequestered in the cytoplasm by KEAP1. Upon oxidative stress, there's a conformational change in KEAP1 that enables the NRF to move to the nucleus where it attaches to the antioxidant response elements with small NAF proteins to unleash a cytoprotective response, many of the antioxidant genes. And we've shown immunohistochemically that there's a biphasic response with NRF2 where we don't see much happening with healthy cells without AMD, but in healthy looking RPE cells, we see nuclear labeling for NRF2, suggesting that there's an increase in activity as a protective response, but when the RPE degenerates, we don't see much labeling. So since mitochondria has been become an interest of ours at being damaged, mitochondrial, we were wondering what cigarette smoke does. And so we started looking at sort of the key different reactive oxygen species. And as most people know, mitochondrial superoxide is one of the predominant ROS that are produced. And when we, sure enough, when we added cigarette smoke to our RPE cells, there's a dose-dependent increase. When we knocked down NRF2 in these cells, there was no augmented response with the NRF2 deficiency. If superoxide is not neutralized, it can react with nitrogen species and form peroxynitrite, which is known to be quite damaging to mitochondria. And we had sort of the same response where there was a dose-dependent increase, but NRF2 deficiency didn't have much of an impact. But when we looked at mitochondrial peroxide after transfecting cells with a plasmid that has a GFP redox specific GFP protein in the mitochondria, we saw that there is a dose-dependent increase, but that there was a marked increase with a further increase in peroxide production with NRF2 deficiency. And so we asked the question, does NRF2 regulate these mitochondrial antioxidants? And just to sort of summarize very simply, superoxide dismutase 2 neutralizes superoxide to peroxide, and then the predominant mitochondrial neutralizer of peroxide is the thyroidoxin 2, predominantly PRX3, which neutralizes peroxide eventually to water, and TRX2 and SRX, which can go from the cytoplasm into the mitochondria, can help to replenish, reduce PRX3 back so it stays healthy and it's able to have its neutralizing effect. And so the long and the short of it is when we looked at each of these different mitochondrial antioxidant genes, both at the mRNA and protein level, there was no real impact of NRF2 deficiency with SOD2, and with TRX2 and SRX, we got the same sort of response where NRF2 deficiency had not much impact, particularly at the protein level. When we looked at PRX3, we got the same sort of flat response. NRF2 didn't have much of an impact, but because PRX3 is predominantly responsible for neutralizing the peroxide, we looked at the oxidized and the reduced forms, and we saw that when we expose cells to cigarette smoke extract, there's an increase, but when we, in our NRF2 knockdown cells, we had a marked increase in oxidized PRX3, which suggested that it was being utilized, oxidized, but was not being reduced back. And that suggested to us that NADPH, which provides the reducing power, is decreased under NRF2 deficiency. So if you think about the mitochondrial sources of NADPH, the most common, the most abundant source is actually in the cytoplasm that comes from the pentose phosphate pathway. But NADPH typically does not cross the mitochondria. So the mitochondria has its own sources. NNT is the most common. It produces almost half of mitochondrial NADPH, but ME3 and GLUD1 are also involved. When we looked at all of these, they really weren't influenced, their abundance wasn't influenced by either smoke or NRF2 deficiency. So that brings us to a little deeper investigation. And so there is some work looking at the isocitrate dehydrogenase shuttle. And basically in the cytoplasm, alpha-ketoglutarate forms isocitrate through IDH1, and NADPH is consumed. And then citrate through the citrate transporter gets transported into the mitochondria, and it starts in the TCA cycle, and IDH2 will then form NADPH. So indirectly, the IDH shuttle can provide NADPH to the mitochondria. And from the literature, we know that NRF2 can regulate both IDH1 and IDH2. And when we looked at if it was affected with NRF2 deficiency, we found that IDH2 was affected, IDH1 was not. And then when we looked at IDH activity with NRF2 deficiency and cigarette smoke, we saw this dose-dependent decrease, but we also saw that the activity was further reduced with NRF2 deficiency. And at the same time, we saw an increase in mitochondrial hydrogen peroxide with IDH2 deficiency. So to provide the NADPH in the cytoplasm, we then went and investigated the pentose phosphate pathway. And there are 3 genes that are highlighted that are NRF2 regulated in the process. And when we looked at all three of them were regulated with NRF2 deficiency. And when we measured NADPH levels, total NADP was actually had a dose-dependent increase with NRF2 deficiency, but NADPH was actually strikingly decreased with NRF2 deficiency. So it suggested that the pentose phosphate pathway was sensitive to NRF2 signaling. So where does that leave us? Just sort of putting it all together, the IDH shuttle with the help of the pentose phosphate pathway can provide NADPH, which is going to provide the reducing equivalents for the TRX2 system to be able to neutralize hydrogen peroxide. And there are various places within this sort of schematic pathway where NRF2 can affect this process. And while we very simplistically thought it was due to the abundance of some of the antioxidant genes, we found it was more related to the production of NADPH. So to provide some relevance, we've done some immunohistochemistry on early AMD samples, and you can see that there's very striking labeling for TALDO1 in the RPE with some decreased labeling in the same patient with either small drusen or larger drusen, and this is just the IgG control. So it gives at least some hint that this process could be going on in disease. So the next question we asked was, how does mitochondrial dysfunction affect the RPE? And what's the consequence of an inadequately neutralized mitochondrial ROS? And so to do that, it's a different project that Sayantan Datta, who's one of our postdocs who has really championed this project. And we've looked at the role of mitophagy, which is needed to repair damaged regions of mitochondria. And basically, if you lose the mitochondrial membrane potential that recruits, PINK1 recruits parkin, there's ubiquitination of mitochondrial proteins, and then you start the whole autophagic process that eventually fuses with a lysosome to break down those products. And we've shown immunohistochemically that PINK1 is decreased in areas of drusen deposits and dysmorphic RPE relative to some healthier areas. And PINK tends to be very strongly labeled in the neurosensory retina as well as the choroid. And when we've looked at PINK1 deficiency, we get the same sort of pattern where we see some mitochondrial superoxide that's increased with either cigarette smoke, and it's a magnified response when we knock down PINK1. And we see a decrease in the mitochondrial membrane potential that's further magnified with PINK1 knockdown. And we see that oxidative phosphorylation is also impaired by PINK1 knockdown, and even more so when you combine it with cigarette smoke. So clearly, mitochondrial function is abnormal. So when we looked at what the impact of the cell was, there wasn't much when we grew cells for 3 days, but by 7 days, a week out, we started seeing that the cell shape became quite elongated. And then we tried knocking down Parkin to make sure it wasn't just specific for pink, and sure enough, we got a similar response. So we did RNA sequencing to look at what the transcriptome was telling us. And one of the striking things we saw is that a number of the oxidative phosphorylation genes were decreased by PINK1 deficiency. But we also saw that extracellular matrix, receptor interaction, cell-to-cell adhesion, and extracellular matrix organization were also affected, that suggested that it was more epithelial mesenchymal transition profile. Phosphatidylinositol signaling was also affected, and I'll get to that in a little bit. So the Broad Institute has a data set of 200 genes that have been associated with EMT, and when we looked at those specific genes, we saw that they were overrepresented, and within those overrepresented genes, NRF2 related genes were also overrepresented. When we went back to our cells and looked at some of the classic markers, we found that a difference in the labeling of E-cadherin here in sort of the red, but also Vimentin also had a redistribution, this elongated appearance in these morphologically altered cells, whereas the E-cadherin label was decreased, and we saw a drop in the abundance of E-cadherin and an increase in Vimentin by Western blot. So to sort of further convince us that these cells were in EMT, we knocked down ZEB1 in our PINK1 knockdown cells, and we could get a reversal of the cell morphology. And ZEB1 is a transcription factor involved in the EMT process. So we then went to a different model, and we used PINK1 knockout mice, and they found that they have a very similar profile where they show impaired mitochondrial function with increased superoxide anion formation, and a decrease in oxygen consumption by the MitoID assay. Beginning at about 4 months, we start noticing that there's an abnormal cell shape on RPE flat mounts, and we've quantified the difference by cell aspect ratio, which measures the diameter, the greatest diameter of the cells. And then in the same mice, we show a very similar pattern of increase in PINK1 or Vimentin and a decrease in E-cadherin. And some of the other, and ZEB1 and snail as EMT transcription factors were also increased, again, beginning at about 2 months, but more prominent at 4 months. And this is similar to work that I've collaborated with Debashis Sinha, where we see evidence of EMT in human AMD samples. So really the next question we asked was, how does dysfunctional mitochondria signal and initiate epithelial mesenchymal transition? And since most of the activity is coming in the mitochondria, we thought, well, maybe it's a retrograde mitochondrial to nuclear signaling that's putting the cells in EMT. And so one of the things, the first experiments we did, we just added chemical oxidants to see if that was working. And it turns out that we got no response when we added tert-hydroperoxide. But if we created the scenario where we knocked down PINK1 and then added mitotempo, which is a superoxide mimetic to neutralize superoxide, or N-acetylcysteine, which works both in the mitochondria and in the cytoplasm, neutralizing mainly peroxide, we saw a reversal of the cell morphology and a drop in ZEB1 expression. We looked at the actual reactive oxygen species and saw there was a commensurate drop as well with the antioxidants. And so as we start to try to figure out this retrograde signaling pathway, we can see that PINK1 is knocked down, but mitochondrial antioxidants seem to neutralize this process, stop this process, as well as some cytoplasmic contribution of reactive oxygen species. So again, we then looked at NRF2 deficiency combined with PINK1 deficiency, and we found that since NRF2 was overrepresented in the broad collection of genes, and when we knocked down NRF2, we expected all the cells to die when they were knocked down with PINK1, and in fact, we saw actually a reversal of the cell morphology. And then to verify that again, we added trigonelline, which is a molecule that prevents the transport of NRF2 into the nucleus, and it had a very similar response. And ZEB1 was also blunted in its expression. And when we went back and looked at the RNA-seq data of comparing NRF2 knockdown to PINK1 knockdown, we saw a very different pattern of AMT genes that were altered by NRF2 deficiency, and that sort of persisted in the double knockdown cells. And then what we did was we crossed NRF2 knockout mice with PINK1 knockout mice, and again we see this abnormal cell morphology in the PINK1 knockouts. The NRF2 knockouts look pretty good at 4 months. The double knockouts look pretty good, and the trigonelline, we got some reversal, a little bit of reversal, but you can see that the main change in the cell aspect ratio when we quantified it was NRF2, or excuse me, PINK1 knockouts, and NRF2 seemed to bring the cell aspect ratio down. And the same thing happened when we looked at the expression of snail and ZEB1 in the PINK1 knockout mice after all these different treatments, suggesting that we're reversing the EMT process. So I like to say beauty is only skin deep. And we asked the question, are PINK1 and NRF2 double knockout cell deficient cells actually healthy? And so when we looked at the cell migration assay, you can see PINK1 cells that are in EMT have a very healthy migratory pattern. The NRF2 knockdown cells, not so much, and the double knockdown cells, not so much. So there's a clear reduction in the health of the migratory ability of these cells. And then when we looked at the cell viability, the double knockdown cells had a marked decrease in cell viability. So the cells seem to be, they look beautiful, but they're not so healthy. And we did an ex vivo experiment where we looked at RPE choroid that we incubated. We just incubated with cigarette smoke, but we just looked at basically the live dead assay, and we showed that again when we looked at the double knockout cells and exposed them for 6 hours in this medium without cigarette smoke, they showed an increase in cell death that's very similar to wild type RPE that was exposed to high doses of cigarette smoke extract, suggesting that they're more vulnerable to dying. So if you go back to our little roadmap here, you can see that if we knock down NRF2 along with impaired PINK1 signaling, we get cell death. So to try to get this link of what happens beyond NRF2 signaling, since NRF2 regulates hundreds of genes, we went to string analysis and we wanted to find any NRF2 related genes that were linked to NRF2 signaling, and we kind of honed in on TXN RD1, and part of that was TXN RD1 was linked to genes related to glycolysis, matrix related, and interferons, which are all connected with the EMT process. So when we looked at TXNRD1, knocking that down in our PINK1 knockdown cells, we were able to reverse that process. And TXNRD2 is related. It's actually mitochondrial located. When we knocked that down, it was very similar, and we got a similar response with ZEB1 signaling. So our roadmap now has identified TXNRD1 as we get toward EMT. And so to figure out a pathway from there, we went back and we looked at our RNA sequencing data and the phosphatidylinositol pathway with PI3K, AKT signaling. When we looked at that, there was a difference in our PINK1 knockdown cells relative to the NRF2 knockdown cells. And you can see that in PINK1 knockdown cells versus control, there's a lot of red dots of genes that were involved that were overrepresented, related to more things in cell cycle, glycolysis, related to mTOR signaling, related to more an EMT pattern. But when we looked at the double knockdown cells, they were related more to cell death with a drop in the AKT signaling pathway. So when we looked at these pathways pharmacologically or by siRNA, we were able to reverse both when we inhibited PI3K or AKT signaling, we're able to reverse this cell morphology of PINK1 knockdown cells. And just we wanted to verify that the signaling process was going on and that we were actually inhibiting the process. And this is by Western blot showing the phosphorylated AKT signaling that we only really see in the PINK1 knockdown cells. And then when we looked at ZEB1, there's a normalization of the ZEB1 levels with the treatment of inhibiting either PI3K or AKT. And when we looked in our PINK1 knockout mice, we get the same thing where we see phosphorylated AKT is markedly increased in the PINK1 RPE of PINK1 knockdown cells, but the other mice with NRF2 knockdown was not present, whereas AKT levels were pretty flat. So this roadmap starts out with impairing the mitochondria that generates mitochondrial ROS that you can neutralize with mitochondrial or cytoplasmic antioxidants, which normally the antioxidants will activate NRF2 signaling to its downstream TXNRD1 that activates PI3K and AKT signaling that activates ZEB1 that eventually induces the cell shape change and then in EMT process. And on the other hand, if you impair NRF2 as well, you're really impairing two different cytoprotective pathways, and that leads to cell death. So again, what we've come up with is a simplified model of RPE heterogeneity, where if you have PINK1 being healthy and NRF2 healthy, the RPE cell is healthy. If you impair PINK1 but have functional NRF2, it can drive the cell into epithelial mesenchymal transition, which is really a cell program to ensure that the cells stay healthy. And that's followed by if you have functional PINK1 but impaired NRF2, they seem to be morphologically normal, but may be susceptible to cell death. And if you have both cytoprotective pathways impaired, you have a much higher cell death rate. And so we think that through impaired mitochondrial dysfunction and other cytoprotective pathways that may lead to some RPE heterogeneity that's well recognized in AMD, and it might help us to understand where things, how the RPE responds to mitochondrial dysfunction. So what I've tried to go through is how to describe how cigarette smoke causes mitochondrial dysfunction and how NRF2 actually had minimal regulatory on the actual mitochondrial antioxidants, but seems to affect more the IDH shuttle and the production of NADPH to indirectly provide NADPH to neutralize peroxide, and that NRF2 deficiency increases mitochondrial peroxide due to inadequately reduced PRX3, and that mitochondrial ROS can lead to mitochondrial dysfunction, but some of the excess reactive oxygen species can be used for initiating a retrograde mitochondrial to nuclear signaling that can lead a subset of cells into epithelial mesenchymal transition, which is part of this emerging story we're trying to play with RPE heterogeneity. And so I thank you for your attention, and I just wanted to point out when I put myself into Wikipedia, I got an error search, and I will stop there. Thank you for your attention.
Thank you, Jim, very much. It was truly wonderful. Any questions you guys have? Jennings.
Yes. So you mentioned in the pink one knockout mice, you saw some RPE dysfunction. I was wondering what the time course was for that, and if you also observed any photoreceptor defects and how the time course for that compared.
Yeah, so for the first question, it's a great question. We actually start seeing mitochondrial dysfunction at about 2 months. So we see it before we actually, so when we've looked at the time course, the RPE cells look morphologically normal until 4 months, but we actually start to see mitochondrial dysfunction at about 2 months. So it precedes the morphologic change. When we've looked at the pink one knockout mice for looking for either photoreceptor loss or any kind of photoreceptor dysfunction, we actually see some mild decrease in the A wave before we actually, and we don't see a whole lot of photoreceptor loss, at least at this point, in unstressed animals. But it's a little bit hard to know for certain because a lot of our work, we're sort of thinking, if we look at the human samples with pink one is very, very strongly labeled through all the layers of the retina. And we only see the differences in the RPE. So the global knockout mouse is not truly representative to understand what the impact of the RPE alone is on it. But it's something that we're continuing to look at as the animals age.
Cool, thank you.
All right, let's move to Greg.
My question has to do, it's kind of a technical question, and it has to do with your cigarette smoke extract. And the question is, how thoroughly characterized is that? And it's a very specific question, is do you know if your cigarette smoke extract has any of these epoxidiols of benzopyrene in it?
So the way it's so we get the cigarette smoke extract from a company in Tennessee, and the way they basically filter smoke through or they blow smoke through a filter, and then they use DMSO to extract it. So it selects more for the lipophilic molecules, and it sounds like it's possible that some of the molecules you're interested might be in it, but it hasn't been comprehensively.
So it probably wouldn't, because in order to get, in order to get these epoxidiols out, so what has to happen is it has to go through the cytochrome P450s. So you get benzopyrene comes out of the smoke, and then it has to go through the cytochrome P450 system to get these epoxidiols out. And the reason I'm interested about that is that those epoxidiols have been shown to be pretty potent NRF2 activators. That's why I was just kind of interested.
Ah, okay. Yeah, I honestly don't know the answer to that.
Okay, Dorota.
Hi, Jim. Very nice to see you again. I have two questions. One is I couldn't help but notice that in your genes deregulated in your models, basically you are getting over and over genes that are deregulated, or there are markers of senescence. And especially EMT transition is quite interesting, because this is a lot of the regulation of the extracellular matrix proteins towards the EMT transition. Did you try to look at this and on other levels? Because on transcriptional level, I think you could find it very easy, but more on some other markers and look at what happens to your RPE cells. That would be really interesting because NRF2 is already known to be involved in the process itself. And then the second question is a little bit more about probably what you are doing right now. It's about the single cell RNA-seq, and question is whether all cells in RPE react the same way. And I would be very interested to see if this is more to cells that are already somewhat stem cells. Did you see them? And so on.
So let me start with the first. So it's interesting you brought up senescence because about 25 years ago, I wrote it was one of the first R01 grants I wrote on senescence of the RPE, and it was not discussed. And it took a lot of wind out of our sails. But you know, we did very carefully try to identify evidence of the senescence program, and we really couldn't see it. And at the time, it was mainly senescence associated beta gal labeling, and the SASP was not really well understood. I have to say that we grew RPE cells out to replicative senescence, and the cell shape is quite different from these cells that look like they're more in EMT, because the senescent RPE cells they have a much flatter, elongated, they look like a big fried egg, right?
Yeah.
As opposed to these more spindly shaped cells. They're both, I think, protective for trying to survive a harsh microenvironment. It's just that we couldn't find any evidence of it. And again, I think certainly oxidative stress can put cells into senescence, but we just couldn't find it in the RPE. I think, to me, immunosenescence might be the place to look, with monocytes, macrophages, whatever, since they have a greater ability to divide. So you have more division, not only oxidative stress. In terms of the single cell kind of work, I mean, it's interesting. We just don't have enough eyes. And part of it is with our work, and I don't know what you guys have gotten, but when we've looked at death to enucleation times of 6 hours or greater, the viability of the cells is pretty compromised. So we've been only getting samples where they're less than 6 hours. And then when the pandemic hit, I haven't gotten an eye in over a year. So what I can tell is that there is a very heterogeneous population of cells, and that was really one of the intentions of doing the single cell work. And there's clear different populations. We call them cells at risk. But we also see cells that are very interesting, where we see an upregulation of a lot of the NRF2 mediated genes. Again, these are early AMD, so there's a protective response. But the thing that drops the most are the visual cycle genes. They drop very, very rapidly.
Thank you. Just two very quick reminders. Many of the presentation in our Friday seminars are posted on the web, so you can watch it if you miss, or if you wanted to hear a second time. And again, if you have any question, please drop me a note via chat, because we're now moving to next question from Chris.
Jim, that was a fantastic overview of the oxidative stress, and it was really very comprehensive. My question would be, if all these components, the NRF2 and the other antioxidant, plays a role for AMD, what are your ideas of why it takes up until the 5th or 6th decade before you start seeing the disease? Is it accumulative? Is it something that's overwhelming? Is it environmental? I mean, what are your thoughts on that?
Yeah, I'm reminded of one of your, I guess, fairly recent publications. So I think the role of epigenetics is important. And if you actually look at the NRF2 promoter, it's very CP rich, CP island rich. So my guess or my hypothesis is that it could be an epigenetic change that eventually blunts it. So we know that if you look, acute smoking will activate NRF2, chronic smoking, at least in our mouse model, it suppresses NRF2 after 6 weeks of daily smoking. So something more chronic, and maybe it's an epigenetic change, we just haven't looked at it. The same thing happens with aging, where just age-related changes can blunt the NRF2 response. So the actual abundance is there, but the ability to respond is blunted. So if we look at a young mouse and we give an oxidative stimulus, there's an appropriate NRF2 response. But if we take an old mouse and do the same stimulus, that response is blunted.
Okay, thank you.
All right, Kathy.
Hi, Jim, that was great. I had a quick question about your pink-1 knockouts and the morphology of your RPE, where they're growing in size. Did you also look at the DAPI or horse staining of the nuclei to see if any of them become multinucleates or anything more than 2, especially in the central retina?
We have not systematically quantified that, but that's something that I've asked Scientin to do because of some interesting work that suggests that the binucleate RPE cells are, some people feel that it's failed cytokinesis as a response to stress. And then some of, I think Chris Curcio's work looking at monkey RPE that are binucleate, the mitochondrial network is much more abundant in these binucleate cells. So it's something to look for, but we just haven't done it at this point.
So when you look in your older ones, it's an easy test, at least, just to stain them with the, and look. But that's, yeah, that's cool. I would expect that to change.
Okay, next will be Henry, Corinne, and Suraj.
Hi, can I go?
Yeah.
Nice talk. This is a little bit maybe off topic, but since you mentioned the NADPH oxidases, they are very important in the superoxide production. And I have a bunch of data sets, sequencing data sets from mice, and they are not expressed in the retina almost at all. At the same time, there are some descriptions that you use selective inhibitors for NADPH oxidases, and they are protective in some retinal degeneration paradigms. I'm a little bit confused how this works. Do you have some insights of these expression levels in the retina and in the RPE, and how high they need to be that there's a biological significance?
I'm not sure I understand your question. You mean in terms of the NADPH oxidases? Is that the abundance? Is that what you're?
Yeah, so I don't find, so basically there's descriptions that show that affecting these enzymes by selective drugs is protective, but I don't see the target in transcriptome level. So I'm a little bit confused, like how this system works.
I'm drawing a blank here. I'm sorry. I'm not really sure. I see the connection. Maybe we can talk offline.
All right, let's move to Corinne.
Hi. Hi, quick question in regards to the in vivo experiments. Have you done or plan to do any visual, like dark adaptation experiments to try to translate that to the human disease in terms of when does it get triggered and how can we rescue it?
The only extent we've done is it has been dark adapting and looking at the ERGs. And it's a little bit, again, it's a little bit artificial because it's a global knockout. We've also done the ERGs at different ages, and we don't see changes until, I'm not sure, 6 months, something like that, when we start to see changes. And then we also, because we have the smoking chamber, we also have mice that are in the smoking chamber to see if we can sort of exaggerate the blunting of the ERG response. But it is a good idea. I mean, one of the things we certainly could and probably should do would be doing scotopic OKT studies to see if maybe there's not so much of a change in light, but we may see more change when they're in the dark. It's a great
Correct.
idea.
Yeah, more important to see if we can protect it, obviously, from happening.
Okay,
Absolutely.
Suraj, you got your turn.
No, I didn't ask the question. Thank you. It's a joy to be here. Jim, that was a very beautiful overview. I was wondering, I was sort of surprised that you didn't see anything in the ER stress area. You said that ER was normal. That is sort of strange because mitophagy is very, very closely related to the communication with the ER. And I was wondering whether did you in your RNA analysis did you look at translocases as to the import function of the mitochondria? What's happening to the translocase expression, which is critically very important for what you are talking about?
Yeah, so the unfolded protein response, so that takes us back a little bit. We had done microarray analysis, and again, we were sort of pointed toward mitochondrial enriched genes. And then when we actually looked at the different arms of the unfolded protein response, I mean, we were very high on that, and we just couldn't find a difference. But again, we didn't go beyond that. Those are only acute experiments. So what happens under more chronic circumstances? I think there's probably some involvement there, but those experiments that I talked about were all more acute. And if you look at what smoke does to the mitochondria, one of the principal things is it actually affects all of the electron transport chains. So you can impair
Absolutely.
1 to 5. So there's probably going to be a quicker response that you see in these acute experiments.
I was curious about Tom 20, Tom 40, Tom 60, the chaperones there, 40 and 60, which are some of them are on the surface of the mitochondria and some of them are in the inner mitochondrial membrane. Was there anything in that area which you found was being impacted? But I thought that would be the place that's where it would start from.
Yeah, we haven't looked at the depth that you probably want us to look at. You know, most of it was just from the RNA-seq data, and we just didn't see anything in the translocases in Tom 20. They were all pretty much the same.
All right, let's move to Dominic, Aisha, and Jim.
Perfect. Thank you.
Dominic.
Dr. Handa, very nice work. Thank you for your presentation. A quick question. You mentioned that you see ubiquitination of mitochondrial proteins in your models. Have you ever checked what proteins are the ones that undergo this ubiquitination the most? And have you looked on the protein level and maybe on the transcriptome level in your RNA sequencing data, potentially?
Yeah, we actually haven't looked at what's been ubiquitinated. It's, you know, I think the slide was just intended to really kind of justify the mitophagy process, but we actually haven't looked at any specific proteins. Is there one that you're particularly interested in?
I was just wondering if you can see some protein that undergoes ubiquitination to the higher extent than other ones that you can actually observe. But if you didn't check it, that's okay. Thank you for your answer. Yeah.
All right, Aisha.
Hi, very nice talk. I have a question. Why you think most of the mitochondria are located in photoreceptors, and yet all the abnormalities you were talking about in your talk is in RPE? What is specific about mitochondria in the RPE versus the one that are most number is in photoreceptors? Why is that? Is there something else that is affecting this whole AMD thing than just mitochondria alone?
Yeah, that's really the $1,000,000 question. I honestly, I don't know the answer to that, but I do. I guess I can offer some potential theories. But first, if you actually look at, I looked very, very hard at a lot of the histopathologic studies to see what's going on with rod mitochondria. Histopathologically, there's just not much that's even commented on in some of these seminal studies. But if you look at the clinical literature with any OCT study that looks at early AMD, there are a number of studies that show that the EZ line, which corresponds with the inner and outer segment of the rods, which is mitochondrial rich, there's actually not a thinning of the line, but there's less of an intensity of that line on OCT, which suggests that there's some alterations in the mitochondria. And those have actually been linked to dark adaptation problems, reduced light sensitivity, some of these same rod-mediated visual dysfunctions. So I think while most of the studies have focused on the RPE, and a lot of the studies that people are looking at in photoreceptors are geared more toward this RPE photoreceptor ecosystem with aerobic glycolysis. But I think the mitochondria are there for at least the EZ mitochondria are there for vision and have very high energy needs. So you need lots of mitochondria, and I think it is affected. And the question becomes: is there, since the RPE seems to be affected first, is there some protective or pathologic signal that the RPE sends to the photoreceptors that either protect or accelerate the rod dysfunction?
Thank you.
Jim.
Yeah, Jim, that was a great talk. Do the cigarette smoke treatments cause DNA damage? And if it does, can that stimulate poly-ADP ribose polymerase activity, which would deplete the cells of NAD and maybe contribute to some of the things that you were talking about at the beginning?
Yeah, it certainly could. It certainly could. We also see, I didn't present it, but we do see a drop in NADPH levels as well.
Yeah. And another question, so when the cells go through this EMT transition, so normally RPE cells express a lot of GLUT1 on both surfaces. And how does the EMT transition affect the GLUT1 expression? Has anyone looked at that?
We haven't looked at it exhaustively. I know that was actually a gene I was interested in because of the whole aerobic glycolysis story that you and others have really looked at. And at least by RNA-seq, there was no difference in our PINK1 knockdown cells compared to controls.
One more question. So, so a lot of organisms, I guess humans even, if you put them in high oxygen concentrations, if they breathe from ventilators or stuff, for high oxygen for a long period of time, it's toxic. And is that true for the RPE cells in culture? If you put them in 95% oxygen, would they get too much oxidative damage? Or if you put them in low oxygen, would they survive better under some of these conditions you're talking about?
Yeah. So remember, all of these studies are done in hyperoxia, right? So the 21% air that we breathe is actually a high oxygen environment. We've actually done studies where we've gone to like 40%, sort of doubled it, and the cells actually go into senescence rather than EMT. So and that's thought to be due to oxidative DNA damage in the telomeres. So.
Yeah, interesting. Yeah, thanks.
Well, let me make a very quick comment. We had, of course, a wonderful presentation today. We're expecting also a beautiful presentation a week from now will be Jim Hurley. So all invited again every Friday, 8:30. And we had a wonderful conversation today. So for final comments, questions, and conclusions, we go to Barb.
Oh, okay. I didn't realize that. I just wanted to make a comment that's not a final conclusion, but I can try and do both. First of all, great work, Jim, as always. I had a comment on possibly answering two questions that people have. One came from Henry asking whether there's any volatile components in your cigarette smoke extract. And the other question was why you don't see ER stress. When we do the cigarette smoke experiments, as you probably remember in our 2014 JBC paper, we showed strong amounts of ER stress in response to cigarette smoke. And what we do is also bubble the cigarettes through the media, but we immediately use the media. We don't freeze it. And you buy yours frozen. So probably a lot of the volatile components are gone by the time you actually get it. And so that might play a role in the differences that we see, that there seems to be a fair amount of ER stress in these cells in response to cigarette smoke extract, but potentially not in yours because you lose a lot of the components of your whatever 4100 or whichever number you had listed that may just be gone by the time you use the media when you freeze it. Jim gave me a wonderful compliment a whole bunch of years ago at ICER, and he said, whenever I start thinking about doing an experiment, and I look in the literature, Barb, you've already done the experiment, and I feel the same way this time around. That I've learned a lot from you in this presentation, and I'm looking forward to your future papers.
Thank you, Barb. I echo what Jim says. So when we actually come up with an idea for mitochondria, I actually do a PubMed search to make sure that you haven't published on it already. My only other response with your comments about the cigarette smoke extract is that I'm still, if you actually look at the papers where we have cigarette smoke extract, I'm very careful on how we describe it as not cigarette smoke or smoking, but as I usually use the term complex chemical oxidant. And the reason for that is we had a paper that was very strongly criticized during the review process because the conclusion of the reviewer was that so few, this reviewer felt that the oxidative stress generated by smoking was not due to the components in cigarette smoke. It was actually due to the inflammatory response and then the inflammatory cells getting to the RPE as the primary source of the ROS production. And there certainly is some precedence in the literature, but by no means is it defined and definitive. So anyhow, I've just been a little bit careful on how we define it. And certainly the way, I mean, you have a lot of volatile stuff, but you have a lot of hydrophilic compounds, and we don't have as much just by the way the smoke extract has been prepared. So yeah, it is going to be different, but it's a complex mixture. And when we compare it to peroxide and some other chemical oxidants, we always get the most dramatic response with the cigarette smoke extract.
Thank you.
All right, well, it was really terrific. Thank you so much, everyone. And of course, most of the thanks goes to Jim. Until next time.
Well, thank you for inviting me. I really enjoyed it.
Thank you. Thank you.